open this academic session in which Mrs. Baidildinova will defend her academic thesis, Platelet Functional Signatures in Venous and Arterial Thrombotic Diseases. I welcome the candidate and wish her lots of success. I welcome also the supervisors, Professor Jurk, Spronk, Professor Wild, and Professor ten Kaat, and of course the opponents and everybody online. But before we start the opposition, I give you the opportunity to tell us something about the subject of your thesis.
Okay, let me share my screen. Can you see it?
Yes. Please start the... Yes, that's perfect.
Okay.
Go ahead.
So, good morning, everyone. My name is Gaukhar Baidildinova. I'm a PhD student from the European Joint Cardio Consortium, and it's my pleasure today to present a summary of my thesis entitled Platelet Functional Signatures in Venous and Arterial Thrombotic Diseases. As it is widely known by each of us, cardiovascular diseases are the #1 mortality reason worldwide. There are two types of blood vessels in our organisms, artery and veins, and thrombus can happen either of them. If thrombi are located in the brain, it leads to acute ischemic stroke. In the heart, it's coronary artery disease, acute coronary syndrome, and myocardial infarction. And I also place here atrial fibrillation as a risk factor for aforementioned conditions. In lower limbs, thrombi leads to peripheral artery disease. Venous thromboembolism describes two related conditions: deep vein thrombosis. It occurs when the blood clot forms in a deep vein, usually in the lower leg, thigh, or pelvis, and a more serious complication called pulmonary embolism, which is an emergency. It occurs when the clot breaks loose and travels through the bloodstream to the lungs. Platelets are known to contribute to pathogenesis of atherosclerosis and subsequent arterial thrombosis, characterized by aggregated but fibrin-clotted platelets and atherothrombosis. And two medications, aspirin and clopidogrel, they block important amplification phases of platelet activation, and they are the first-line treatment for these conditions. However, many patients do not react to such treatments as expected and would still develop thrombotic events. It can be explained by high treatment platelet reactivity, and the prevalence of this has been estimated to 5 to 60%. And please pay attention to this term, HTPR. I will refer to this several times during my talk. Once again, HTPR is so-called resistance to the antiplatelet treatment. When platelets are activated, they release various pools of molecules. Altogether, they are called as platelet releasase, and it comprises three types of biomolecules that are proteins, peptides, and small molecules. The protein releasase, by other classification, divides into secretome, sheddome, and extracellular vesicles. Molecules secreted by platelet granules are called secretome. The main sources here are alpha granules. Sheddome is a set of molecules that are shed or proteolytically cleaved from the platelet surface, like membrane, proteins, or receptors. And there are also extracellular vesicles, which are lipid-borne vesicles released by platelet into the plasma. On the right side, you see an activated platelet, and we see that chemoattractive proteins like beta-thromboglobulin, STL1, and platelet factor 4, they are released from alpha granule. On the other hand, receptors such as GP5, PLEG2, P-selectin, and CT40 ligand, they are shed from the platelet surface to enhance communications with the leukocytes and regulate platelet activation. Then we also see the small molecule thromboxane A2, which is a potent positive feedback mediator during platelet activation. And it also enhances the platelet activation. And this process comes from arachidonic acid via enzyme COX-1, and this is blocked by aspirin. So my PhD aimed to provide a comprehensive understanding of prothrombotic and pro-inflammatory platelet activation markers and functional signatures in arterial vascular diseases and venous thromboembolism. My thesis comprises 6 chapters, but due to time limitations, I will focus on chapters 4 and 5, which are representative for my thesis. I will start with chapter 4, written based on patients from Maastricht cohort. And first, let me explain you the study design and our aim. So we had 105 patients with peripheral artery disease. They were recruited in Maastricht. 60 out of them, they were on aspirin, 45 on clopidogrel, and patients who received both treatments were excluded from our analysis. We also pooled them as a third group to increase the patient number, having as a result 105 patients. And then we took whole blood from these patients with citrate as anticoagulant, and platelet aggregation was measured in specialized machine called Verify Now to measure platelet reactivity to arachidonic acid to evaluate aspirin treatment, and reactivity to ADP to evaluate clopidogrel treatment. Furthermore, we generated plasma platelet pool plasma from this blood and quantified proteins using a proximity extension assay, determining 184 cardiovascular related proteins. Because the Verify Now related units for aspirin and clopidogrel detection differ, the different values and units for both Verify Now tests were also standardized and integrated as a new continuous variable to enable a combined analysis of the Verify Now data of both aspirin and P2Y12 inhibitor users within the pooled group. And then we quantified, and then we looked at association between Verify Now values and protein expression in plasma using a distinct biostatistic method, the random forest algorithm. So we wanted to see in the end what proteins are associated with HTPR or resistance to aspirin and or clopidogrel. And here you can see the patient characteristics at baseline, stratified by antiplatelet therapy. So the first column, all patients, the second column is aspirin group, and clopidogrel 45. The therapy adherence among patients was high, and both antiplatelet groups, they were comparable in terms of sex, age, BMI, and hypercholesterolemia. Also, the percentage of MI history, diabetes, and smoking, and they were similar between aspirin and clopidogrel groups. There were more patients with a history of stroke in the clopidogrel group compared to aspirin. However, more patients on aspirin therapy had prior revascularization. So here you can see the main results from the Baruter and the forest plots. So in aspirin group, we found 3 upregulated plasma proteins that was significantly associated with higher treatment platelet reactivity to aspirin, meaning we detected 3 proteins that are responsible for so-called resistance to aspirin treatment. In case of clopidogrel treatment group, these proteins were 6, and in the pooled group, we found 10 upregulated plasma proteins. Then we wanted to discover more about the function of these proteins. So for this, I performed a protein-protein interaction network analysis. As you can see, 3 plasma proteins from the aspirin group, they did not result in any clustering, meaning they have distinct functions and are not involved in any common pathways. In contrast, 2 interconnected clusters with 2 interacting proteins in each clusters were created for 4 out of 6 proteins associated with HTPR for clopidogrel. And these proteins, they participate in innate and adaptive immune responses. And in total, you could see that here 8 out of 10 proteins selected for HTPR in the pooled groups, they were connected to 1 cluster. These proteins are involved in coagulation, atherogenic dysfunction of blood cell vascular interaction, and cell death. This elevated number of proteins in this common cluster may be due to increase in patient number in the pooled group. So our conclusion is that plasma protein signatures, they may serve as determinants for poor platelet responsiveness to aspirin and clopidogrel in peripheral artery disease. And on the right side, you see the cartoon created by me where we placed all the involved proteins. If you want to know more details about them, you're welcome to read our articles in thrombosis research. And we are moving to the chapter 5 with individuals from the Mainz cohort. So again, study design and methods. So here we have a subcohort of population-based cohort in Mainz. It is called the Gutenberg Health Study, and we had 903 participants that were randomly recruited between 2012 and 2022. These participants with a mean age 62 plus minus 11 years were not only healthy people, but also presented with frequent cardiovascular risk factors, cardiovascular diseases, and comorbidities accompanied by the treatment. As you can see, I performed several function assay, but here I will focus only on LTA or light transmission aggregometry in full. So LTA measures the percentage of platelet aggregation in response to various agonists that activate platelets and make them aggregate. Obviously, the percentage goes from 0 to 100, and in response to small concentration of weak agonists, as we also used, you would expect a low aggregation percentage. But that wasn't the case. Platelets from some individuals would still aggregate quite strongly, and we were interested if this phenomenon is associated with some clinical determinants and genetics. But first, we wanted to see what is hyperreactivity to define it. So in our analysis, we used a subthreshold concentrations, meaning low for ADP and epinephrine. It was 0.5 micromolar. And we plotted all the values for aggregability, and you could see two Gaussian distribution. So one for low aggregability in blue and one for the high aggregability in red. And their cross point was set as a threshold. So for ADP, it was 52%, and for epinephrine, it was 73%. Then we defined four groups to compare with each other based on whether they are low or high responsive to ADP and epinephrine, but for the sake of time, I will demonstrate only two comparisons. If you're interested in all four, please read our articles published recently in Blood Advances. So first, how high ADP responders are different from ADP low respondents in terms of clinical determinants and genetics. And the threshold, as I told, was set at 52%. So you see what is high responders, low responders. And second, epinephrine high responders versus epi low responders, the threshold is 73%. So what did we find? Utilizing robust Poisson regression analysis, estimated prevalence ratio revealed that atrial fibrillation and dyslipidemia are related to ADP high responsive platelets. And in case of epinephrine high responsive platelets, we found an association with a history of venous thromboembolism and again atrial fibrillation similar to ADP. Based on the analysis of 56 common variants of SNPs related to platelet phenotype and function, two variants in the gene of GP6 representing an important platelet collagen receptor, they demonstrated predictive impact for increased platelet ADP aggregability. While the MPYG6B variant and gain-of-function variant in PR1 showed predictive impact for platelet high reactivity to epinephrine. You can see they are here presented on the platelet. Conclusions. So evaluation of platelet ADP and epinephrine high responsiveness might provide predictive value for enhanced platelet activity in atrial fibrillation, dyslipidemia, and venous thromboembolism. In summary, Chapter 4 described the establishment of system-based plasma protein signatures associated with high treatment platelet reactivity in peripheral artery disease, which may be helpful in identifying alternative, novel, and personalized treatment strategies. In Chapter 5, atrial fibrillation, venous thromboembolism, dyslipidemia, and common variants in GP6, PR1, and MPG6P may have a potential impact on platelet high responsiveness to ADP and epinephrine. The impact of my thesis is that it provides new insights into the potential fine-tuning of antithrombotic management to improve the efficacy and safety balance in the individual patient with thromboinflammatory cardiovascular diseases. And as acknowledgement, I'm grateful to many people who supported me through this thorny past. First and foremost, my biggest thank you goes to Dr. Kirsten Jurk, who is a dream PhD supervisor and without whom I would most probably quit my PhD. Further, I would like to thank my supervisors, Professors Hugo Tunkate and Philip Wild, and the great support by Dr. Vincent Tenkate. Also my family and closest friends as Claudia Schönichin, Dr. Oksana Yurikova, and others. And now I'm excited to answer your questions.
Thank you very much for your nice presentation. We start the opposition with the chair of the assessment committee, that's Professor Heemskerk. He is Emeritus Professor of Cell Biochemistry of Thrombosis and Hemostasis in the University of Maastricht. Professor Heemskerk.
Yeah, dear candidate, my congratulations with this thorough thesis. I have read it with great pleasure, especially the well-structured general introduction and discussion. And in my congratulations, I'll also like to extend to your supervisory team. So I do have some questions, and maybe it will not a surprise to you that my questions will relate to chapter 5. There, it was nicely illustrated in your presentation. You define threshold levels of high activity or low activity of platelets from a group of healthy controls and several patients with cardiovascular diseases. And then, if I understand it well, in the final end, you then conclude that there is a difference between the healthy subjects and the patients. So my question is, why didn't you do it the other way around? Just compare the healthy subjects with the patients and then conclude something on the difference of platelet activation. So why did you choose for this approach to draw your conclusions?
So yeah, apparently it started like we are more interested in platelet function, and we could see this phenomenon that some individuals, we would say healthy kind of individuals, I mean not healthy, but like general population cohort, they have this increased aggregation for platelets, and we were interested if it's connected to some clinical determinants, and probably in the future it could serve as some biomarkers to predicting probably some clinical outcomes. So yeah, rather than going backwards, probably it would lead us to some other direction we wanted to see. Yeah, this way around.
Okay, so then let's if I go to page 227, this is also a figure that you showed in the presentation. Then you say, I'm looking at the ADP response. You say that now at the epinephrine response, you say that the vast majority of the subjects is a high responder. So there I wonder if I read your conclusions well, is it right that many of the high responders then indeed are patients rather than healthy subjects?
Yes, yeah, highly esteemed opponent. So as I told, we had patients with a mean age of 63 years, plus minus 11. So it would be like kind of more elder people, and plus they're not like purely healthy. It's like based on the general cohort. I mean like real cohort. So they also had some cardiovascular diseases and determinants. So probably this can explain why they're like more epinephrine high responders, as you would probably expect. So I think probably if it would be younger generation with like less disease burden, it would be low amount of epinephrine high responders.
Okay, so that brings me to the question: how was this threshold determined? Was that determined?
Yeah, we placed all the values for the hyperreactivity, and we could see these two Gaussian distributions. And then we decided that the lower one apparently kind of like demonstrates people with like normal reactivity or low reactivity, and the second was hyperreactivity. And where they crossed, we placed the threshold is here. So I think in our case it was quite obviously seen.
Okay, so you did not, I mean, I would have expected that you first will do a threshold on a healthy population and then determine what the effect of the patient is. That is not what you did.
Yeah, because all of the thresholds, what is like hyperreactivity and so on and so forth, it was mainly arbitrary. So we wanted to have more objective approach to this. That's why we, started with some statistical method, which gives like more robust definition for all of this. What is hyperreactivity and so on.
What is your mechanistic explanation for this hyperactivity? What is different in those people, healthy subjects, patients, where you see this hyperactivity?
So
About that.
we saw that in case of ADP, these patients, they have atrial fibrillation and dyslipidemia. So patients, not patients, individuals with these diseases, they are more prone to have hyperreactivity platelets. And in case of epinephrine, it would be again atrial fibrillation and history of venous thromboembolism. And there were also several SNPs determining this effect. I mean, still like fortunately these people, they're still alive, so we didn't have a chance to correlate with clinical outcomes, but probably in like 10-20 years we would see if they correlated with this and to make like more robust conclusions out of this.
So you related to the pathology. I was also thinking on let's say biochemical factors, because it is known that both the ADP response and epinephrine response are sensitive to desensitization, and you can get either positive or negative priming of platelets. Have you thought about that kind of mechanical platelet signaling dependent explanation for this difference? Something with ADP levels or so, or?
Yeah, I mean, epinephrine compared to ADP has more pleiotropic effects. So it's not only activate platelets and lead to platelet secretion, it also can lead to muscles contraction. So for example, even to intestine contraction and apparently to the heart rate. So for example, if you inject epinephrine, it led to transient atrial fibrillation. It was demonstrated by 2 studies. So because this is more pleiotropic, I believe it had like first a higher percentage of people, and then this effect. But since they also like kind of correlate with each other. So for example, we also looked at other agonist and LT values. We could see that if patients are like individuals hyperreactive to one agonist, they would also hyperreactive to other agonists. So it also indicates that there is like a concomitant effect between them. So their biochemical pathways kind of crossing, not like kind of, they're crossing. Yeah.
Okay, so I have a last question, if I may.
No, I'm very sorry. I'm very sorry, Professor Heemskerk, but we're running out of time. Thank you for your opposition. The following opponent is Dr. Depperman. He's also a member of the assessment committee, and he's an associate professor of the Center for Thrombosis and Hemostasis in the Johannes Gutenberg University of Mainz, and it's in Germany. And thank you, Dr. Depperman, for coming all the way from Germany.
Thank you, Pro-Rector. Dear candidate, thank you very much for this very clear presentation of your work. I have a few questions about chapter 3, where you are looking for platelet-derived plasma factors. And among them, you find P-selectin and CD40L. And my question here would be, are they really platelet-specific, these markers?
Thank you, esteemed opponent, for your questions. You're talking about the chapter with venous thromboembolism, right? So indeed, yes, P-selectin and CD40 ligand, they are also secreted from in the thelial cells as well. But for example, I also did a review on this. This is chapter 2, and actually, how it states in articles, like 95% of circulated P-selectin comes from the platelets, and also it's correlated with platelet number. So we would assume that it's rather of platelet origin, but to make it more robust, it would be good to also concentrate on platelet-specific proteins like soluble DP5, DP6, and so on and so forth. But we kind of assume that it's more of platelet origin, based on the literature.
Thank you. Just a follow-up question on that. So do you think it's soluble P-selectin, or do you also, like with your isolation method of your plasma, do you still have vesicles, extracellular vesicles in there, right? Do you still have them in there?
I mean, plasma apparently would have extracellular vesicles, but we didn't measure them. We measure more like soluble plasma. Yeah, the method picks up soluble proteins.
Okay, but is it spun down at really high G with the ultracentrifuge to get rid of them, or would you think they're still in there?
I mean, to get rid of them, you would need like really high centrifugation speed. So I would assume that this, yeah, I didn't think we did this. So I assume extracellular vesicles would be still there.
Okay, thank you. And then I have my next question is about figure 2 in the chapter 3, and I assume there's a lot of data behind this figure, and it's just too much data to show. So it's like all condensed in this heat map. Could you elaborate a little on what is on the data that is condensed in that figure to make it a bit clearer for me? I mean, I'm not used too much to look at these kind of figures. So maybe you can just elaborate a bit, because I think there's like hundreds of whatever proteins in there, and it all comes down, condenses in this color. Yeah.
Exactly. I mean, the message of this figure 2 is rather simple. So we wanted to differentiate these patients to 3 groups: DVT associated PE, isolated DVT, and isolated PE. And first, we wanted to see if their protein profile is indeed different. So that's why it was did a heat map, and since like you see that colors are quite different between these 3 groups, it confirms that this 3 group of patients indeed have different distinct protein profiles, and it makes sense to go deep in this. If they would be the same, then we would probably say, okay, these phenotypes are not that much different between each other, at least in terms of platelet profiles. So yeah, it looks complicated, but the message is simple, that they're different, have distinct protein profiles.
Okay, thank you.
Yeah, because many clinicians still question if it's like 2 separate the states of the disease, or it's the same, and so on and so forth. So yeah, we proved this from the terms of biochemistry, that they're different.
So that these are really different entities.
Yeah, exactly.
Yes, wonderful. Thank you. My next question is about the technique called lasso regression. Could you maybe explain us a little bit what you're doing there and what you use it for?
So yeah, lasso regression was used to see what kind of proteins would be associated with these conditions and which of them are platelet related. So yeah, this was the aim. Yeah, this is basically what I can say about statistics, because it was performed by our statisticians, so they are more experienced. I just understand the general implication of this. Yeah.
And then you basically relate them to make sure that they are platelets, that they are derived from the platelets, because then you get this association, right? That's
Yes, exactly. We want to see that they are of platelet origin, and then we also did additional analysis to see if they correlate with this platelet markers, like typical markers.
Okay. One other finding that you have is that you find ACE2 in your plasma samples, and again, I think that's the soluble version then of the receptor, and that leads to you having or finding a pathway that is called pathogen entry. So how does that relate? How is that connected to the diseases that you are studying here?
Yeah, I mean, probably some of the complications were caused by viruses. So, for example, AC2 is kind of like famous receptor for COVID-19, which is apparently caused by the virus. And then it also leads to complications in terms of thrombosis. So, yeah, it also confirms that these conditions can be caused by like viral origin. Probably, yeah, I would assume. So it was like, yeah, interesting to see that ACE2 here is present among our proteins.
Okay, thank you.
You're welcome.
Thank you very much for your opposition, Dr. Depperman. Then we come now to the following opponent, and that's also a member of the assessment committee. That's Dr. Warlee. And Dr. Warlee is an associate professor of vascular surgery at the Radboud University Medical Center, and that is in Nijmegen. And thank you for coming all the way from Nijmegen, albeit online. Dr. Warlee.
Thank you, Mr. Pro-Rector. Dear candidate, first my congratulations for your very nice thesis with solid papers in it. Also, I would like to involve your whole supervising team in my congratulations. First, I would like to discuss table 4 in your introduction, where you present a very nice overview of all the genetic variables, gene polymorphisms, in genes affecting aspirin and clopidogrel function. Especially for clopidogrel, which is a prodrug, the carriership of CYP2C19 which is allele 2 or 3, is a loss of function allele. So in these patients, the prodrug is simply not converted into an active metabolite. So that's a quite easy way to predict the function of the drug. Wouldn't it be much easier to put our money on and further investigation on genotyping instead of these difficult platelet function assays? For simple clinicians like I am.
Dear esteemed opponent, thank you for your compliments and your questions. I mean, genotyping is also not that much cheap. So yeah, probably it makes sense for us to assess how much this polymorphisms are present in the population, which is a common in the patients what you are treating. And if it's rather high, then yeah, to see if it's like connected to outcomes. But since the prevalence of this HTPR, so resistance to the treatment, is rather high, if further investigations would confirm that it's indeed helpful, I would probably go for the genetic genotyping. But yeah, first it needs to be proved that it really makes sense and it's highly prevalent among patients.
I agree with you. Then I would like to go to chapter 2, where you do not study gene variants nor platelet function, but you choose to study soluble platelet release factors. So, in my view, these factors may be even a more indirect measure of platelet function. So why did you choose to study these soluble platelet release factors?
Because they are rather kind of like easily accessible, because you basically just draw blood from the patients and then you can apply this PEE method, or basically it's combination of ELISA and PCR. So it's also kind of like it's robust methods. It shows you numbers, and if it confirmed that it's distinct between patients, then it could be used routinely. So that's why we were interested in the measurements, yeah, because it's easily accessible and kind of like established routine.
Would these soluble factors replace platelet function tests?
No, currently, unfortunately, it's still rather on experimental stage. So I would not say they are replaced. We need further to evaluate if they correlate with each other and if they're connected to clinical outcomes to make any concrete conclusions out of this. So I would keep doing this in combinations with each other.
So there might be an additional value in a prediction of combining platelet function tests with soluble factors.
Yeah, I think we should keep doing this. And for example, if we talk about diseases that are related to platelet disease, so for example, von Willebrand factor disease, von Willebrand disease, or Bernard-Soulier disease, they are detected by platelet function test. So yeah, I believe it should still be used in combinations. I mean, for experimental purposes, not like on regular routine clinical base.
I agree. Then I would like to go to chapter 4. In vascular surgery, in patients with peripheral arterial disease, we just prescribe clopidogrel, which is first choice, and not aspirin. This choice is based on a large trial 2 or 3 decades ago, the Capri study, in which clopidogrel was more effective in preventing arterial thrombotic events as compared to aspirin. In figure 2 on page 193, you did the proteomic analysis comparing patients on aspirin with clopidogrel. And to my surprise, what I see is that in patients on clopidogrel, 9 inflammatory proteins are increased. So to me, this does not explain the better efficacy of clopidogrel as compared to aspirin in preventing adverse thrombotic events in PAD patients. How can you explain this?
How can I explain this? Yeah, I mean, the purpose of this figure was to see if, indeed, again, similar to the previous chapters, the proteome of these patients is different between aspirin users and clopidogrel users, because if it would be the same, then it wouldn't make sense to see it. So in clopidogrel users, yeah, indeed, you see increase of several proteins. I think, I mean, probably you would use clopidogrel as a first line, but from what I read, I may be wrong because I'm not clinician. They are used together, and sometimes the strategy to defeat this HTPR, I mean, resistance to clopidogrel, would be used to higher doses. So probably in that case, yeah, in that case, clopidogrel need to be used in higher doses to be better. But yeah, I don't think this figure shows that clopidogrel doesn't help. It just shows that they have different profiles.
Yes, I agree. Why did you choose to relate your proteomics to disease severity and not to the occurrence of thrombotic adverse events?
Very short answer, please.
Yeah, we didn't have these thrombotic events that many. That's why we couldn't correlate with them. That's why we chose rather disease severity.
And you only had 9 patients on HCPR, right?
Okay.
6.
Okay, thank you,
With the-
Mr. Prorector. I'm satisfied with the answers.
Thank you very much, Dr. Vale, for your opposition. Now we come to the following opponent, that's Professor Falanga, who's also a member of the assessment committee, and she's professor of hematology in the University of Milan, and that is in Italy. Thank you for coming all the way from Italy.
Thank you. Thank you. Dear candidate, thank you for your presentation. Congratulations to you and your supervisors for the work you have done for this thesis. Well, I would like to focus on chapter 3, on the relation of platelet activation with venous thromboembolism. The aim of your study was to identify plasma protein signatures related to platelets to distinguish VTE subtypes, specifically to distinguish the phenotype of PE, either isolated or associated with DVT, from DVT alone. So I am asking, what is your expectation from this distinction you found in your results? Do you think it can help and how in the management of these patients?
Dear highly esteemed opponent, thank you for your compliments and the questions. Yes, I mean, first, people until the kind of recent times and still probably would question if platelets in general participate in the venous thromboembolism pathogenesis. There were several studies confirming this also on mice, in vitro, in vivo, and so on and so forth. So our article first confirming that yes, indeed, platelets are involved. So as Hugo said, so for example, when doctors are not sure if these patients have arterial thrombosis or venous thrombosis, they would give them antiplatelet medications together with anticoagulants. But then when they discover it's rather venous thromboembolism, they would cancel antiplatelet treatments. But if we see that platelets are involved, then apparently you need to continue with them. So probably in the future, some protocols will be changed, and then since the platelet contribution is different between these entities, it means that should be different approach to this again. So like rather than DVT and PE, they should not be treated in the same way. Yeah, but again, would be better to correlate with some clinical outcomes. that would come up, fortunately, in 20, 30 years. I mean, fortunately for them, unfortunately for us researchers. Yeah.
So you think that the DVT alone can be in the future, according to your results, also distinguished as far as the treatment from T associated or not with
Exactly.
DVT? Okay,
Yes.
I am satisfied. Thank you.
Thank you. Thank you.
I have no other questions.
Okay, thank you.
Okay, thank you for your position, Professor Falanga. Then we now come to the following opponent, that's Professor Dijkgraaf, who's Professor of Biomimetic Chemistry in the University of Maastricht. And today she has also accepted the role of Secretary of this committee, for which I'm very grateful. Professor Dijkgraaf.
Yeah, thank you, Pro-Rector. Dear candidate, first of all, I would like to congratulate you on the completion of your PhD thesis. It was very educational for me and also a joy to read your thesis, so my compliments for that. And of course, I also would like to include your promotion team in my congratulations. But of course, I do have some topics which I would like to discuss with you. And first of all, and this is a bit related to the questions my colleagues, Professor Vangela and also Dr. Depperman have asked you already. And yeah, it's about proposition number 1, because this one really caught my attention. It's not necessary to read it out loud, but there you are discussing the inclusion of newer plasma markers of platelet activation. There I saw one of the chemokines in this row as well. And when I go to chapter 3 on figure 3 and also figure 5 on page 135, there you give platelet-related proteins which you could use as biomarkers. Which one would you choose? Because there's quite a list. Which one would you choose if you would like to set up a test? If we have a look at page 135.
Highly esteemed opponent, thank you for your compliments and questions. I would start probably with a platelet specific markers like GP6, GP1B alpha, SD1 alpha, GP5, and CLEC2. This would be my choice rather than going for all. But if you have extra money, then yeah, you could include more. But this would I listed my first choice because they are more platelet specific.
Yes, I agree with you, because I was really wondering, because if I have a look in figure 5, there I see that CXCL12 or SDF1 alpha is upregulated and downregulated in either DVT or IDVT. Would this be a suitable marker or one of the other chemokines like CXCL5 or CCL7?
Yeah, probably. I mean, the thing is, when I looked at these papers, people first measure them mostly in patients with arterial thrombosis, and not that many articles I found related to venous thromboembolism, and then also data were inconclusive. So yeah, if they would just include more, and the chemokines you listed also could be candidates. Yeah, but I wouldn't go crazy and include all of them. I would start with a small amount of proteins to measure.
Yeah. We discussed this already. Indeed, you would go for the more platelet-specific biomarkers, because this was also what really caught my attention, because I know the chemokines, and especially SDF1 alpha or CXCL12 from cancer. So so I really thought, well, would this be a suitable biomarker for DVT or IDVT or other thrombotic disease? And this is also what you mentioned in the discussion in your chapter. Let me see, where was it? Yes, on page 140, it's the first paragraph. There you say that our cohort of patients, and there you have the exclusion of active cancer. How did you do that? Because in my opinion, a malignant oncologic lesion is always active. And how did you demonstrate that these cohort patients did not have active cancer?
It was confirmed by clinicians, I assume their test, but definitely this cancer patients needs to be excluded, because if they have cancer, then all the like inflammation and other markers would be increased, and we wanted to see of the effect of specifically venous thromboembolism rather than what is caused by cancer. And yes, indeed, these markers can be increased not only in case of venous thromboembolism, arterial thromboembolism, it also can be increased in Alzheimer's disease patients, in also immune patients, and so on and so forth. But it doesn't mean that, how to say, if they increased in our patients, we can say, oh, probably it's like prognosis, a marker for worse outcomes, worse diagnosis, and it can be the overlap with other diseases as well. So
Yeah, exactly.
we don't,
Yeah.
It's
So
not like,
yeah, please go ahead.
yeah, I mean, it's like the same with inflammation or high temperature. It's we don't ignore the symptoms just because they are present in other patients. We still need to consider them if we want to see if it's differentiate between patients with better outcomes and with worse outcomes.
Yeah. Okay, but to come back to the question about the exclusion of active cancer, actually the clinicians did this. So you don't know whether they made a PET scan before patients were included in the study or whatever. You're not familiar with that, or?
I'm not quite familiar with the procedure, but I believe they ask them first, and then active malignancy is checked by several tests, like you do if there are like oncocells are present or absent, and stuff like this.
Yeah. Okay.
You send them probably to a CT or MRI.
Yes. Yeah, there are various methods indeed to visualize whether people have cancer or not. Because to come back to this CXCL12, it's highly upregulated in various tumors, and it's really a chemoattractant for metastases, and especially to the lungs. And I was wondering whether this can also be one of the chemokines when I read your thesis, and it might be nice to investigate this further. Because it's a platelet activator as well, that it causes also lung embolism in the end over there, because that's what you usually see with cancer patients, and they usually also form thrombi. They have usually already a thrombotic event beforehand, before they are diagnosed with cancer. Okay, but having said that, I was wondering if you would go to the clinic with your biomarkers, what kind of technique would you use to analyze the presence of the biomarkers? And I'm focusing on the proteins, and what kind of test would you use?
Probably I would use ELISA. I mean, PE is also a good method. But I believe it's probably more expensive than ELISA. It depends on how much they cost. But first we need to prove if they really indeed correspond to clinical outcomes before we apply this into the clinic. Yeah.
What if you would go to LCMS methods? Would that be an option for you?
LCMS, you mean mass spectrometry?
Yes, mass spectrometry, so that you analyze it first on HPLC and then mass spectrometry?
Yeah, I mean, before you go to the clinic, you need to be sure that everything is confirmed on the like experimental stages. So I think it's a good approach to also try not targeted proteomic methods, because we applied like distinct panels, and they apparently can have only limited number of proteins. And when you apply mass spectrometry, you can detect all of them, but it's kind of like measures all the types of protein expression, and then you need bioinformaticians to detect this, to calculate. But I think it should be combined together with this before you go to the clinic. But when it's confirmed, when you have like ready panels, then you can go with ELISA and PCR.
Yeah, I fully agree with you. I fully agree with you on this, because especially if you have proof of concept testing, then it should be fast, and then ELISA is much faster. I'm happy with your answers, and I give the word back to the pro-rector.
Thank you, Professor Dijkgraaf, for your position. Then we go to the following opponent. That's Dr. Kuipers. and she's a scientific researcher at the Department of Biochemistry in the University of Maastricht. Dr. Kuipers.
Yes, dear candidate, I would also like to take this opportunity to congratulate you on this beautiful dissertation. I read it with great pleasure, and I also would like to include your supervisory team and your family and friends in my congratulations. Of course, I also have a couple of questions because, yeah, such nice work also triggers a lot of questions. But I first would like to start actually with the title of your thesis and also your general aim, because it's all about platelet functional signature. But to me, it was not really quite clear what you actually mean with a platelet functional signature. What is your definition of this?
Esteemed opponent, thank you for your compliments and questions. So platelet functional signature, we wanted to measure and see the function of the protein that associated with platelets, to see how it looks in patients who have different types of arterial thrombosis types and venous thromboembolism, and to see what is the function of this protein. That's why it's called platelet functional signature. So it's not only one protein, rather it's signature, because one protein probably is non-informative, so it's better to use them in combination. That refers to signatures, and to see in which pathways they are involved. That's why it's functional signatures.
Okay, yeah, thank you. So you already indicate indeed that probably to get a really meaningful platelet signature, you need to look at multiple proteins, and you all have also done this in your thesis, in all your chapters, looking at the different diseases. But I was wondering that if you really want to use such a signature for in a clinical setting later on, and it also kind of relates to what Professor Dijkgraaf already mentioned, is that you need to measure all these proteins, and what then, and if you need to really have a good solid signature, you need to combine several markers. But you can, of course, not measure all of them because it would be really very expensive and also take a lot of time. So what would you then focus on, and do you need to use only the markers, but can you also take into account some functional data, or for example platelet count? Is it also important to include this in such a signature? Can you elaborate a little bit on this, how you envision how many features do you need to measure?
Yeah, I mean, platelet count is always there from the blood analysis before we approach with panels. Yeah, of course, first you need firmly to prove that they're indeed clinical relevant, because yeah, I had the same question from my students actually, who are medical students, and I told it's all experimental. First, you need to prove that they indeed are related to some clinical outcomes. And then, for example, with venous thromboembolism, we proved that the platelets are involved, but probably it does make sense to count these proteins for them. But probably in case of PED patients, so if we measure them and we see that this protein is probably present, and we could assume that they have this tendency to HTPR, so they would not react to neither aspirin or clopidogrel, then probably you can use other anti-platelet treatments, probably need to increase the dose of clopidogrel and so on and so forth. So first, yeah, you need to define your aim for what you want, and before you come to this, you need to be sure that they are relevant to these clinical outcomes. Platelet counts is easy to add, and yeah, the rest probably some like point of care tests like Verified Now, and probably LT, which is also golden standard, and yeah, proved to be certain and kind of like equal between labs. So yeah, I would go for Verified Now and LT if you need. Yeah, but not to go directly with all of that.
Okay. Yeah, I think I agree with you. I think you need to look at multiple things, and for also to really best help each individual patient. I also have some questions about another topic, because nowadays more and more it's becoming clear that one platelet is not similar to another platelet, and that there are different platelet populations. You have so-called aggregating, secreting, and procoagulant platelets, which also have been shown to differ between different diseases. And then there's also a thing called the age of the platelet. So I'm wondering if you think that these different platelet populations and also platelet age play a role in these platelet signatures. Do they influence the signatures that you see?
We didn't look specifically at the subtypes, but we make an analysis adjusted for the age of patients and sex and many other variability which could have influence. And we had two models, like model 1, just age, sex, and platelet count, and with other medications, and they did not differ much. So I would assume that these factors don't have that much effect in the end.
Okay.
Because the results we got were quite similar between each other.
Yeah, but I actually mean not really the age of the patient, but as you know, a platelet can circulate in the human body from 7 to 10 days. And there are differences between like young platelets, which have just been released from the bone marrow, as compared to platelets which already circulating for 10 days. Do you think this also affects the platelet signature, the things they release?
Yeah, as I told, things like other factors did not influence much, like taking of anticoagulant medications and so on and so forth. So probably the age of these platelets per se also don't make that much sense. And I would assume, I don't think it's much difference. I mean, probably we can do additional analysis to see if it's differences, but like this, I wouldn't think so.
Okay,
Yeah.
well, that's good to know. Then you don't have to take that into account. And I was also wondering, why did you choose to measure actually all these markers in plasma? Because you actually measure platelet markers. And would it not be better to actually look at the platelets then of these patients to really study different proteins that are different? And do you think that if you would have also looked at the platelets of the patients, that you might have also found other biomarkers?
Yeah, probably the set could be different, because in plasma, it's not only platelets, it's also other proteins coming from other blood cells. But I think in the clinic, it's easier to look at the plasma, because for this, you just need to centrifugate the blood rather than purification of platelets. So I think this would be just closer to the clinical application compared to pure platelets.
Yeah, you're right. It's more easy to separate plasma, and I think also plasma you can store in the freezer. Platelets don't really like that. So I understand for practical reasons that you have to look at the plasma. I'm looking at the prorector. Can I ask some more questions? Because I also prepared some questions about chapter 3.
Yeah, go ahead. There's still time.
Yes. So in chapter 3, you measured proteins in different groups of PE, pulmonary embolism, which are both isolated or which are either isolated PE or DVT related PE, and then you compare it to a group that only has DVT, if I understand correctly. And that you find that these platelet-related proteins are different between the groups. You show this in figure 5 on page 135. And then you show also that the platelets, the proteins that are released from these platelets can be released via different granule secretion or shedding or extracellular vesicles, as you also very nicely presented in your presentation. But I wonder, what is actually the trigger for platelet activation in isolated PE versus DVT-related PE? Do the platelets have a different trigger to activate them in these different diseases?
Yeah, probably. I mean, isolated pulmonary embolism would just have less serious conditions.
You may finish your answer if you want, shortly.
Yeah, I think isolated DVT is less serious condition. That's why it doesn't have this pool which comes together with deep vein thrombosis. That's why it's different. Would be my guess.
Okay.
Okay. Thank you, Dr. Kuipers, for your opposition. Ms. Candidate, Mrs. Baidildinova, the time appointed for the defense of your thesis has passed, and the degree committee will now withdraw to discuss the quality of your thesis and the way you defended it. I ask you and your company online to await the results of our deliberation and our return. I suspend this meeting.
Mr. Prorector, you are muted.
Ah,
Yes, you're unmuted now.
yeah, okay, I reopen this academic ceremony. Mrs. Baidildinova, the degree committee here present online has assessed the quality of your thesis and the way you defended it. And in view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. And Professor Ten Kate is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite Professor Ten Kate to now take the floor.
Thank you, Prorector. It starts with a promise that is required. Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
Thank you. By the authority vested in us by law and in conformity with the decision of the committee here present online, I hereby confer upon you, Gaukhar Baidildinova, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, you will soon receive the degree certificate signed by the rector, the secretary, and the supervisor affixed with the official seal of the university, shown by the beadle. And let's see, she does show it. Thank you very much, beadle.
Thank you. Thank you.
You're welcome. And first of all, I'd like to thank all the co-supervisors and the attendants to the ceremony for their presence and their critical questions. And well, you did well, Gaukhar.
Thank you.
It's for me now also to read a few lines as laudatio. And I went back to your application before you came to Maastricht, where you first started, in the application letter from 2019, where you wrote that you had obtained a bachelor degree in biotechnology at Al-Farabi Kazakh National University in Kazakhstan. It's far away. We've never been there. Maybe some of the committee members, but I haven't been yet. Additionally, you studied for 1 year the master course at the same university, being accepted with 198 points out of 200. So you were quite smart. That's important. After the first year of master course at this university, you deregistered from your university for studying at University College of London. And you wrote, and that was quite interesting. I think for you, it was quite a challenging experience. Also a unique opportunity to study there, thanks to a special award that was granted to you by the President of the Republic of Kazakhstan in 2015. And at UCL, you chose a project related to neurobiology and studied the accumulation of amyloid beta plaques in the hippocampus. So really far from clotting and platelets. And maybe not so much, after all, in mice of different transgenic lines. Since my course, as you said, since my course was a Master of Research, I spent much time in the laboratory doing immunohistochemistry, working with the microscope and software. And importantly, you said, I have never studied before in English. And it was only 12 months for adaptation, doing research and writing the thesis. So it was a very challenging period in your life. The first months, you didn't even understand the people around who were speaking to you. My English language skills did not allow me to comprehend different accents, especially the British one. And that's a tough accent indeed. Therefore, my master course was a huge challenge for me. But in December 2016, you obtained your diploma with merit from the University College of London, and I'm extremely proud of this achievement, and you can be. That's what you said. I cite this little story because it probably illustrates your search for the better and the beautiful in life and work. We know that living in Kazakhstan, like in many foreign countries, is not easy for young academics with ambition beyond a family life. You were a teacher in between, and you also enjoyed being that, as you mentioned during the defense. I quote one more and last time from your information, where you said, I was a private tutor since my 21st years, and I'm a teacher at the University nearly for the whole year. I adore teaching. I believe I can change their mind of the students to give them unique knowledge and also inspire them, how I was inspired by my teachers before. This combination of apparent talent, motivation, and ambition and competencies made you a great candidate for the PhD position in the T-Cardio consortium. Your English supervisor stated that you had a quick intellect, and that argument convinced me. It sounded appealing. You started in Maastricht in the biochemistry department, and for many reasons, this was not the easiest stay. I can summarize it as there was not so much chemistry overall. You worked on a very nice and important review paper with Paola van der Meijden on soluble platelet activation markers, and this work is well cited. The experimental work did not fly well for many reasons, and only when you arrived in Mainz to join the CTH and work under the supervision of Professor Philip Wild and under the careful guidance of Professor Kerstin Jurk, you became more comfortable. Kerstin and also Vincent Kate, the younger one, have supported you consequently and with great care is my impression, also from your acknowledgement. The product of your stay in Maastricht and Mainz is this impressive thesis, and you can again be proud that you completed this work and obtained a doctorate degree. Life and work in different countries and moving back and forward from Asia to Europe is extremely challenging, and it's a pity that obtaining a visa is just so awfully complicated, no matter where you are, unfortunately. It's such a pity that you could not manage in a reasonable manner to get a visa in order to defend your thesis here, but it's not your fault, it's bureaucracy. Finally, as Professor X, from your acknowledgement, I'm very pleased that you made it. I regret the initial troubles in Maastricht, but congratulate you also on behalf of Dr. Paola van der Meijden and Professor Henry Spronk. I'm most grateful for the continuous support of my Mainz colleagues. I want to say I really much enjoy and support our collaboration and hope it will last for many years ahead. I also thank the European Union, the Horizon 2020 program for making the T-Cardio program possible, and hope and expect that we will be involved in a new program. With that, I wish you, Gaukhar, your relatives, family and friends a good health and a bright and better future without fear and without war. Thank you.
Thank you so much, Hugo.
Thank you, Professor ten Kate, for such a beautiful laudation. And dear Dr. Baidildinova, also on behalf of the Maastricht University, I congratulate you with the degree you have acquired. And I extend my congratulations, of course, to your supervisors, Professor ten Kate, Professor Wild, Professor Spronk, and Professor Jurk, and also your family, friends, and colleagues. I hope you can celebrate it in a good way, and I wish you a very successful further career. I would like to thank the members of the opposition for their presence and their work, and of course the PADEL and the Maastricht University support team, the technical support team, who made this online PhD session possible. Having said that, I now close this academic session.
Thank you.
Okay, so this is not streamed.